So in the previous slides, it was shown how the replication of DNA makes it possible for our new cell to contain the same genetic information as its parent cell. So how the genetic information contained in a cell is expressed in cell operation will now be considered. So this leads to the topic of the protein synthesis. So the synthesis of proteins is for the skin, the hair, enzymes, hormones, and so on. So it's under the direction of the DNA molecules. Okay, so it is this role of DNA that establishes the similarities between the parent and its offspring that are regarded as hereditary characteristics. So protein synthesis is divided into two phases. We have the transcription and the translation. So this is the general process for the DNA synthesis. It is starting from the DNA, and this will undergo a transcription process to convert into RNA. Then the RNA will undergo the translation process to produce a protein. So let's just define or talk about first the RNA, the ribonucleic acid, because this is already related to the protein synthesis. So these are the four major differences between the RNA and the DNA. So the sugar unit in the backbone of the RNA is ribose. However, in DNA, that is the deoxyribose. So the base thymine found in DNA is already replaced by uracil in RNA. So the combination or the partner of the adenine in RNA is already the uracil. So RNA is a single-stranded molecule. Take note on that. While DNA is double helix, meaning it is double-stranded. Thus, RNA, unlike DNA, does not contain equal amounts of specific bases. So RNA molecules are much smaller compared to the DNA molecules, ranging from 75 nucleotides to a few thousand nucleotides. So there are types of RNA molecules, so we're going to enumerate each of them. First is the heterogeneous nuclear RNA. It is using this abbreviation HNRNA. So it is an RNA formed directly by the DNA transcription. So post-transcription processing converts the heterogeneous nuclear RNA to messenger RNA. So messenger RNA or mRNA is a RNA that carries instructions for protein synthesis, that is the genetic information, to the sites of the protein synthesis. So its molecular mass varies with the length of the protein whose synthesis it will direct. Third, we have the small nuclear RNA, that is snRNA. So it is a type of RNA molecules that facilitates the conversion of heterogeneous nuclear RNA to a messenger RNA. So it contains from 100 to 200 nucleotides. Fourth, we have the ribosomal RNA. So it is abbreviated by small r and RNA. So this is a type of molecules that combines with specific proteins to form ribosomes. So already know that ribosomes is the site for the protein synthesis. So the physical sites for protein, again, synthesis that found in the cell is again the ribosome. So the rRNA present in ribosomes has no informational function. Lastly, we have the transfer RNA or the tRNA. It is a type of RNA molecules that delivers amino acid to the sites for protein synthesis. So, of all the types of the RNA molecules, tRNA is the smallest, possessing only 75 to 90 nucleotide units. So, This is an overview of the types of RNA in terms of cellular locations, where they are encountered and processes in which they are also involved. So this is in the cell nucleus. This is the process and the molecules that found, and this is on the cytoplasm. So as you can see, DNA replications found in the nucleus, also the transcription, then on the, what do you call the boundary between the nucleus and the cytoplasm, we have the mRNA. And the translation process is occurred in what? In the cytoplasm to produce the protein. So as you can see, this is the process for the protein synthesis. So in the cell nucleus, we found heteronuclear RNA, sRNA, and mRNA. Then here we have the rRNA and the tRNA. So at a non-detailed level, a cell consists of a nucleus and an extranuclear region, that is the cytoplasm. So the process of DNA transcription occurs in the nucleus, as does the processing of the H and RNA to mRNA. So DNA replication also occurs again in the nucleus. Then the mRNA formed in the nucleus travels to the cytoplasm. That's why mRNAs can be found both on the cell nucleus and on the cytoplasm. So this just summarizes the transcription and translation process in terms of the types of RNA involved and the cellular locations where the processes occurs. So let's proceed with the transcription process. So transcription is the process by which DNA directs the synthesis of the H and RNA, mRNA molecules that carry the coded information that is needed for the protein synthesis. So gene is a segment of a DNA strand that contains the base sequence for the production of a specific H and RNA, mRNA molecule. So in humans, when we talk about genes, most genes are composed of 1,000 to 3,500 nucleotide units. So hundreds of genes can exist along a DNA strand, obtaining detailed information concerning the total number of genes and the total number of nucleotide base pairs present in human DNA. So it was an area of intense research activity during the years of 1980s to the 1990s. So there are already many research conducted with regards to the genes. Okay, so when you say genome, is all of the genetic material, which is the total DNA that contained in the chromosomes of a certain organism. So I think when we say genome, we already heard some projects that related to the genome, just like the genetic engineering, many experiments conducted with regards to this one, genome and genes. So these are the steps in the transcription process. So first, a portion of the DNA double helix unwinds, exposing a sequence of bases, a gene. So the unwinding process is governed by the enzyme ribonucleic acid polymerase rather than the DNA helicase or the replication enzyme. So DNA helicase is also known as replication enzyme. So free ribonucleotides, one nucleotide at a time, aligned along one of the exposed strands of the DNA bases. So the template strand forming new base pairs. So in this process, uracil rather than thymine aligns already with the adenine. So in the base pairing process, only about 10 base pairs of the DNA template strand are exposed at a time. So because ribonucleotides rather than the deoxyribonucleotides are involved in the base pairing, ribose rather than deoxyribose becomes the incorporated into the new nucleic acid backbone, because we're already talking about RNA. So the base must be, the pentose sugar, sorry, must be already ribose. So third, RNA polymerase is involved in the linkage of the ribonucleotides, one by one, to the growing hnRNA molecule. So transcription ends when the RNA polymerase enzyme encounters a sequence of bases that is a read as a stop signal. So the newly formed hnRNA molecule are the RNA polymerase enzyme RNase, and the DNA then rewinds to reform the original double helix. So this is just the summary. And again, in DNA RNA base pairing, the complementary base pairs are, so for DNA, we have adenine, guanine, cytosine, and thymine. Then for RNA, we have uracil, cytosine, guanine, and adenine. So RNA molecules contain the base uracil instead of the base thymine. So the strand of DNA used for the hnRNA mRNA synthesis is called the template strand. So this is the meaning. So this is the template strand. So it is copied proceeding in the 3 to 5, this one, 3 to 5 and direction. So the other DNA strand, the non-template strand, is called the informational strand, this one. So the informational strand, although not involved in the RNA synthesis, gives the base sequence present in the hRNA strand being synthesized, with the exception of uracil replacing the thymine. So this figure shows the transcription of DNA to form hnRNA involves an unwinding of portion of the DNA double helix. So again, only one strand of the DNA is copied during the transcription process. So this is just the summary for the transcription phase. Again, step 1 is the formation of the hnRNA, wherein the DNA in the nucleus partially unwinds to allow a strand of hnRNA to be made, just a one strand only. Then step 2 is the formation of the messenger RNA. Introns are removed from the hnRNA strand. Then lastly, mRNA enters already the cytoplasm, in which the mRNA also leaves the nucleus and enters the cytoplasm part of the cell.